Welcome to the Deep Dive.
Great to be here.
Today, we're taking a deep dive into some recent developments in prostate cancer research. We've pulled together insights from a stack of sources covering everything from findings in clinical trials to some really promising new work happening in the lab.
Yeah, and our mission for this deep dive is really to distill these sources into the most important findings for you. We want to give you a clear, sober look at some potentially new avenues for treating this disease, covering quite a range of different approaches.
Okay, let's unpack this then. We'll start by looking at some updates from clinical trials, maybe beginning with therapies that are in, later stages of testing.
Absolutely. Makes sense. So, one interesting set of findings involves an immunotherapy candidate called CN-2409. This is being looked at for newly diagnosed localized prostate cancer, specifically in patients with intermediate to high-risk disease.
CAN 2409. And this one, it's received some notable designations from the FDA, hasn't it? I see Regenerative Medicine Advanced Therapy, or RMAT designation. And that was after an earlier fast track. What does that actually signal to you?
Well, these designations, they indicate that the FDA sees real potential here, potential for this therapy to address an unmet medical need. And, they're looking to potentially speed up its development and review process. It puts it on a faster track, which is, well, pretty significant.
Right. And that RMAT designation, it's supported by a pivotal phase 3 trial, I believe.
Yes, exactly. It was a randomized, placebo-controlled trial, quite large, 745 patients. They combined CAN-2409 with velociclover and standard external beam radiation therapy.
Okay. And the results from that trial, how promising were they looking?
They were very promising, actually. The trial met its primary endpoint. It showed a statistically significant 30% reduction in the risk of prostate cancer recurrence or death compared to the placebo group, the ones just getting standard care.
Wow, 30%.
Yeah, and there was also a 38% reduction in what's called prostate-specific disease-free survival risk.
A 30% reduction in recurrence risk. That sounds pretty substantial. What does that really mean for a patient with, localized disease?
It's a big deal. Reducing the chance of the cancer coming back by nearly a third is a major step forward. For these patients, it could mean delaying or maybe even preventing the need for more intensive treatments later on.
Right.
So it offers a potentially better long-term outcome.
Okay. What about PSA levels? That's something doctors track closely.
Right. So the therapy significantly increased the proportion of patients who achieved a very low PSA level. Specifically, they looked at a nadir below 0.2 nanograms per milliliter.
Okay.
And that was 67.1% in the CN-2409 arm compared to 58.6% in the control group. And having a low PSA nadir after treatment, that's often associated with better long-term control of the disease.
And looking directly at the tumor itself, what did the 2-year biopsy show?
Good question. At that 2-year mark, 80.4% of patients in the CN-2409 group had what's called a pathological complete response.
Meaning no detectable cancer.
Essentially, yes. No detectable cancer remaining in the biopsy samples. That compared to 63.6% in the control group. And importantly, the safety profile looked consistent with earlier studies, too.
How does this therapy actually work? It sounds quite different from just standard radiation.
It is different, but it's designed to work with the radiation. Basically, it uses a modified virus, a replication-defective adenovirus, to deliver a specific gene, one from the herpes simplex virus, directly into the tumor cells.
Okay.
Then, when you give the patient oral valciclover, which is a common antiviral drug, it gets converted inside those tumor cells into a substance that kills the cancer cells nearby. But, and this is key, this process also stimulates the patient's own immune system, specifically their CD8 plus T cells, to recognize and attack the tumor.
Ah, so it's kind of teaching the body to fight the cancer from the inside out.
That's a good way to put it, yeah. It's generating an immune response against the tumor itself.
That's a really fascinating approach. Okay, let's shift focus a bit. Let's talk about metastatic castration-resistant prostate cancer, or mCRPC. So this is cancer that's spread and isn't responding well to standard hormone therapy anymore. We've got data here on a type of treatment called Radioligand therapy, specifically 1727 Lu-PSMA-I and T.
Correct. And this data comes from the first prospective multi-center Swiss registry study. That's important because it gives us critical real-world evidence, not just strict clinical trial data.
Okay.
Now, 177 Lu-PSMA-I and T, it targets PSMA. That's prostate-specific membrane antigen, a protein found on the surface of many prostate cancer cells.
And who was included in this registry study? What kind of patients?
It enrolled 333 consecutive mCRPC patients. These were individuals whose cancer had already progressed on standard treatments. Many were what we call heavily pretreated, meaning they had already received multiple therapies like AR signaling inhibitors, maybe chemotherapy.
Right, so a challenging group. What did this real-world study actually show then?
It demonstrated both safety and efficacy, which is what you want to see. The safety profile, side effects like effects on blood counts, kidneys was quite similar to the established radioligand therapy, 177 Lu PSMA 617. You might know that one as Pluvicto.
Pluvicto. Yeah.
And most of the side effects were mild to moderate.
And how well did it work? Did it control the cancer?
The efficacy results were definitely encouraging. 41% of patients saw a partial PSA response during treatment. That means their PSA levels dropped significantly. And 17% actually achieved a PSA 90 response, a drop of 90% or more.
Wow, okay.
Looking at survival, the median overall survival was 13 months, and the imaging progression-free survival was 6.5 months.
So given it seems pretty similar to Pluvicto in terms of safety and shows decent efficacy, does this suggest 177 Lu PSMA-I and T could be another solid option for patients?
Yes, absolutely. It provides more evidence for the effectiveness and safety of PSMA-targeted radioligand therapy in this mCRPC population, and it could definitely serve as a valuable alternative, maybe depending on availability or specific patient factors.
Okay. Sticking with mCRPC for a moment, we also have new data from an early trial, a phase 1b for a drug called Oracy 944. This was tested in combination with standard androgen receptor inhibitors like apalutamide or darolutamide.
Right. This trial specifically looked at patients who had already progressed on prior AR pathway therapies, so again, a group needing new options. They tested Oracy 944 combined with one of those AR inhibitors.
And what kind of response did they see with that combination?
They saw what's described as broad and deep PSA responses, and that was across all the dose levels they tested. Some key numbers. A 59% PSA 50 response rate, so over half the patients had their PSA drop by at least 50%.
That's quite high.
It is. And a 24% PSA 90 response rate.
Were those responses lasting? Sometimes you see a drop, but it doesn't hold.
That's a crucial point. And yes, importantly, the report says the responses were durable. Several patients actually remained on the treatment for nearly a year or even longer. So that suggests the combination could provide some long-lasting benefit for certain patients.
Okay. And Oracy 944, it's positioned as a specific type of inhibitor, isn't it?
Yes, it's being developed as a potentially best-in-class PRC2 inhibitor. That's the target. And in this early trial, the combination regimen seemed generally well tolerated. Most side effects were mild, grade 1 or 2, with the most common being some low-grade diarrhea.
All right, let's look at another radioligand therapy now. This one's being explored in advanced solid tumors, and that includes prostate cancer. It's 177 Lu FAP 2286, part of the ongoing Lumiere study.
Yes, this one's quite novel because it targets something different. Fibroblast activation protein, or FAP.
FAP, okay.
Instead of just targeting the cancer cells directly, FAP is found on supportive cells within the tumor's environment. These are called cancer-associated fibroblasts.
So it's hitting the tumor support system, not just the tumor cells themselves. Is that the idea?
Exactly. The therapy uses a small peptide designed to find that FAP protein, and it delivers lutetium-177. The researchers describe this as creating a crossfire effect. The radiation hits the FAP-expressing support cells, but it also damages the nearby cancer cells, too.
Interesting approach. How do they figure out if a patient is actually a good candidate for this FAP-targeted therapy?
Patients are screened beforehand. They use gallium-68, FAP, 2286, PET, CT scans. These scans can show if the tumors and where they are actually express FAP.
Makes sense. And what do we know so far about, say, safety and how the radiation is distributed in the body? It's still early days, right?
It is still early, but the initial data suggests the therapy is well tolerated, mostly mild side effects reported.
Yeah.
Dosimetry studies, which measure where the radiation goes and how much, are also important here. They're showing the radiation does concentrate in tumor sites. For example, bone metastases, which are common in prostate cancer, absorbed a mean dose of 3.0 gray per gigabecquerel, while critical organs, like the kidneys and bone marrow, seem to receive much lower doses, which is what you want for safety.
Good. Okay, now let's shift again slightly to metastatic hormone-sensitive prostate cancer, or mHSPC. This is a cancer that spread but is still responding, at least initially, to standard hormone therapy. We have a phase 2 clinical trial here for something called Luxdegalutamide, also known as JSB462.
Luxdegalutamide.
Hmm.
Yes, this is part of a really exciting newer class of drugs called proteolysis targeting chimeras, or PROTACs.
Protacs. I've heard about these. How do they work differently?
Well, they're designed to work very differently from traditional hormone blockers. Instead of just blocking the androgen receptor signal, a PROTAC is engineered to actually grab onto the androgen receptor protein itself and tag it for destruction. It basically uses the cell's own garbage disposal machinery to get rid of the receptor protein entirely.
Wow, okay, so it removes the target.
Exactly. And importantly, this approach might even work against resistant forms of the AR receptor that can develop in advanced disease, mutations found in up to maybe 24% of mCRPC patients.
And this phase 2 trial is testing it in combination therapy.
Yes, it's a randomized, open-label, multi-center study. It's testing luxdegalutamide taken orally at 2 different doses combined with abiraterone, and they're comparing that against standard AR pathway inhibitors like abiraterone or enzalutamide given alone.
Are there any early hints about how well this might work, maybe from related trials?
Yes, there are some insights from a related phase 1/2 trial. That one was in the mCRPC setting. It showed promising results, specifically a 43% PSA 50 response rate in patients who had known AR mutations, like the L702H mutation, which is known to cause resistance to some standard AR therapies.
Okay. And side effects?
Common side effects in that earlier trial were reported as generally mild things like fatigue, some nausea, vomiting.
So the potential here is its ability to actually degrade the receptor, possibly overcoming resistance mechanisms, plus it's an oral treatment.
Hmm.
Could be significant advantages for these mHSPC patients, perhaps?
Absolutely. It's a really novel mechanism, and testing it earlier in the disease course in mHSPC is a logical next step.
We should also touch on another combination approach, this one back in mCRPC, specifically for patients who have certain genetic mutations. These are called homologous recombination repair mutations, or HRRM. This data comes from the TelePro-2 trial.
Ah, yes, Talapro-2, combining Talazoparib, which is a PARARP inhibitor, with enzalutamide.
Right. So explain HRR mutations and why this combination makes sense.
Okay, so homologous recombination repair is a critical pathway cells use to fix damaged DNA. Some cancers have mutations in the genes involved in this pathway, like maybe BRCA1 or BRCA2.
Like in breast cancer sometimes.
No, exactly, same genes often. When these HRR genes are mutated, the cancer cells become more vulnerable to drugs that target other DNA repair mechanisms. PARP inhibitors, like Talazoparib, are designed to do just that. So the idea is a synthetic lethality hitting 2 pathways at once is lethal to the cancer cell.
Okay.
The TALAPRO-2 trial specifically selected patients with these HRR mutations, identified using a 12-gene panel.
And how did the combination perform compared to just giving enzalutamide alone in these HRR mutated patients?
For the overall group of HRR mutation patients in the study, the combination significantly improved several key outcomes. They saw better response rates, longer radiographic progression-free survival, meaning how long until the cancer grew on scans, and importantly, improved overall survival compared to just enzalutamide alone.
Were there specific HRR mutations where the benefit was maybe more pronounced?
Yes, they did break that down. The benefit seemed most significant in patients with mutations in BRCA2. But significant benefits were also seen in patients with CDK12 mutations. Patients with ATM mutations also showed notable improvements. Those with CHEK2 mutations, the benefits were a bit more modest, but still there. It really highlights the growing importance of doing that genetic testing to figure out who might benefit most from these targeted combination approaches.
Yeah, that personalization aspect seems key. Okay, so we've covered quite a bit of ground on the clinical trial front. Let's move now to work happening even earlier in the preclinical stage, before human trials.
Right. And there's particularly novel drug candidate here moving closer to clinical trials for mCRPC. It's called QED-203, and it's being developed out of the University of Queensland.
QED-203. What makes this one stand out? Why is it novel?
Well, it's described as a first-in-class small molecule. That's because it targets a mechanism that really hasn't been exploited before in prostate cancer treatment. It focuses on a specific protein called TRPV6.
TRPV6.
Yeah, researchers are actually calling this an oncogenic because it's found at high levels in prostate cancer and other cancers too, and it seems to act like a channel that fuels tumor growth.
And QED-203 is designed to block this channel.
Precisely. Early lab studies show it very effectively blocks this TRPV6 channel, even at very low concentrations. And blocking it seems to disrupt key signaling pathways that the cancer relies on.
What does that disruption do to the cancer cells?
It seems to trigger what's called endoplasmic reticulum stress inside the cancer cell. It halts the cell cycle, stops the cells from dividing, and ultimately pushes the cancer cells towards programmed cell death, that apoptosis we mentioned earlier.
Okay, telling them to die off. How did it perform against cancer cells that had already developed resistance to other common treatments? That's always a big question.
That is really key. And the preclinical data here looks quite promising. It showed that QED203 retained its power, its effectiveness, against cell lines that were resistant to standard drugs like enzalutamide and darolutamide.
Even the resistant ones.
Yes. And even more impressively, it was reported to be effective against cells that had the problematic ARV7 splice variant. That's a known and difficult mechanism of resistance to AR therapies. In these resistant models, QED203 actually demonstrated superior potency compared to the standard drugs.
And what about in animal studies? Moving beyond cell lines.
In mouse models of prostate cancer, the drug appeared to be well tolerated, and it was effective at inhibiting tumor growth and also reducing PSA levels. The results were shown to be comparable to enzalutamide in those specific mouse models.
So, thinking big picture for QED203, as a non-hormonal agent targeting a completely new pathway, this could potentially offer a fresh option, especially for patients whose cancer has become resistant to the standard AR pathway therapies.
That's exactly the hope, yes. It represents a fundamentally different way to attack the cancer cell itself, moving away from just targeting the androgen receptor pathway.
Okay, so we've looked at a lot of different approaches today. When we try to connect the dots from these different sources, what's the big picture look like to you?
Well, I think it really highlights the intense and very diverse research efforts that are underway to tackle prostate cancer from multiple angles. It's not just one strategy. We've seen promising approaches that try to harness the patient's own immune system, like that in situ vaccine SAN 2409.
Right.
We've looked at targeted radioligand therapies delivering radiation precisely to cancer cells marked by PSMA, or even, as we saw with 1987 LUFAP 2286, targeting the tumor's support structure by going after FAP.
Yeah, the crossfire effect.
Exactly. Then we're seeing totally new drug classes emerging, like the PRTACS Luxdegalutamide being an example designed to actually degrade the very protein driving the cancer, even the resistant forms.
Destroying the target.
We also talked about exploring combinations of existing drugs to leverage specific vulnerabilities, like those HRR mutations, using PARP inhibitors with AR inhibitors.
Telepro too.
Correct. And finally, we're seeing the identification of completely new targets that weren't previously drugged, like that TRPV6 oncochannel, with candidates like QED203 coming through the preclinical pipeline.
So each approach seems designed to overcome specific challenges, wouldn't you say? Whether it's improving control of localized cancer early on, or finding new ways to treat advanced disease that's become resistant, or even personalizing treatment based on the cancer's specific genetic makeup or protein expression. It really feels like a multi-pronged attack on a complex disease.
It really is. And the overall goal for all of these is ultimately to improve both efficacy, how well the treatments work, and safety or tolerability. The aim is to provide more effective options, hopefully options that can be tailored to individual patients at different stages of their journey and with different biological forms of prostate cancer.
It's certainly clear that the fight against prostate cancer is seeing continuous progress, which is encouraging. Researchers are exploring such a wide range of innovative therapies. This deep dive into just some of the latest findings gives us, and hopefully you, valuable insight into these ongoing efforts to develop more effective and more targeted treatments.
Absolutely. And what's particularly striking, I think, is the increasing understanding of the specific molecular vulnerabilities within prostate cancer cells and also within their surrounding environment. And that deeper understanding is really what's paving the way for these potentially more personalized and hopefully more impactful therapies in the future. It's about finding the right treatment for the right patient at the right time.
Stay strong and fight on.